Good evening, everyone. Kazue Patrick once more, and today I'm going to discuss with us the developmental anomalies. So, what are developmental anomalies? Developmental anomalies can also be referred to as malformations or developmental defects, and these are abnormalities of the structure and function that occur during the developmental stages of an organism. And they are characterized by the absence, deformity, deficiency, or excessive growth of body tissues or organs. So, before we can continue on, we need to understand teratology and the associated concepts because they are a key factor in the cause of these congenital anomalies. So teratology, it's a division of embryology and pathology involving the study of the abnormal developments and congenital anomalies. So somebody might ask themselves, why are we seeing now teratology, embryology, and pathology? So when the teratogens, which are the factors causing defects, when they exert their effects in the stages of development, what we call embryogenesis, these effects can lead to malformations or improper development, or they can disrupt the formation of certain structures in the body. And when this occurs, it can lead to a disease state, which is pathology. So that's why teratology is a common term that can be exchanged as we are studying embryology and pathology. Then teratogenesis is simply the process of how these defects arise, especially during embryogenesis. Then we can understand the principles of teratology. These are 6 principles, and the first one goes susceptibility to teratogens will depend on the genotype of the conceptors and the interaction with the adverse environmental factors. So this means that those conceptors whose genotypes are unstable and are exposed to adverse environmental conditions can easily become susceptible to the teratogens, and they will eventually easily develop the anomalies. The second principle is that susceptibility will vary depending on the developmental stage at the exposure, and the exposure is to the teratogens, whereby the most sensitive stage is the stage of embryogenesis or organogenesis. So this is the stage whereby the embryo is beginning to develop the important organs, the important tissues within the body. And this period is between the 3rd and 8th week of gestation. So during that period, teratogens can easily affect the development, leading to anomalies. The third principle is that teratogenic effects depend on the dose and duration of exposure. So this means that the higher the doses of the teratogens and the longer the periods of exposure to these teratogens, the more susceptible will be and the more likely the outcome of anomalies. Then the fourth, teratogens will act in a specific way on developing cells and tissues. So this means that they target specific tissues and organs within development, and they will act by either disrupting cell division, differentiation, and tissue formation. So that's where they will exert their specificity in action. Then the fifth principle is that the interactions of the genetics and environment can result in complex outcomes, meaning when there is a possibility of the genetics, for instance, if they are unstable genetics of a given conceptors and the environmental factors are extreme, the combination of these can result into diverse congenital effects, which are most times lethal. And this summarizes that genetic susceptibility, maternal health, and environmental conditions are key factors in bringing about these congenital anomalies. Then the sixth and the last principle is that manifestations of abnormalities depend on the developmental stage at exposure. Simply, we can say this is timing. So, when the teratogens are exposed in the early stages of development, they will lead to structural malformations. Then when the exposure to the teratogens is in the late stage of development, this can result to functional or behavioral defects. Etiology of the malformations. So this simply means the cause of these malformations. We have to note that the causes of majority of the malformations are idiopathic. So it means that these malformations, it is very hard to trace what exactly is their cause. But the following are some of the factors that have been able to gather that are really associated with most of these malformations. The first is the genetic factors. So these genetics, especially when they interact with the environment, they can readily bring about defective genes. These defective genes can be inheritable, or even they can be like mutations. And these can be changes in the number and the structure of the chromosomes. And this is evident in the cases of Down syndrome and the Klinefelter syndrome. The second cause can be exposure to the teratogens. Remember, we said that teratogens, they are the factors that can cause the defects. And these teratogens, they can be infections, especially the intrauterine, because that's where the fetus is developing in the uterus. They can be poisons and environmental toxins, or even medications. The other cause of congenital abnormalities is the nutritional imbalances. So these can either be deficiencies or excesses of certain nutrients, especially nutrients like vitamin A and vitamin B9. We very well know that these play critical roles in the development through influencing organ formation and tissue differentiation. So their deficiencies can lead to malformations such as the eye malformations, where we shall have microphthalmia, the case where the eyes will be small, and ophthalmia, where there will be no eyes at all. Then we can also have craniofacial abnormalities like cleft lip and the cleft palate. We are going to view these more in the next slides. Then we have incomplete closure of the spinal cord, which is known as spina bifida, and then anencephaly. The other cause is hyperthermia, that is elevated or extremely high temperatures. So these high temperatures will interfere with the protein synthesis, especially of the proteins known as the heat shock proteins. These proteins act like bodyguards to our body, which will respond in terms of stressful events such as extreme temperatures, extreme colds. So if we interfere with their normal synthesis, it means we shall lack these bodyguards. And this will lead to disruption of our membranes, the cellular membranes, leading to cell death. We can have vascular disruption because the vasculature is also made of cells. We can have placental infarction, having destroyed the vasculature, the cells. So, and in turn, all these will lead to damaging of the developing cells, thereby leading to the abnormalities. Now we shall move on to the general presentation of the malformations. So what do we see really for any given malformation? This is what we can be able to characterize or simply find out about it or observe about it. So there will either be arrest of development, where we shall see the development having stopped somewhere, it has not reached the full capacity. For instance, in the cases of aplasia, agenesis, or hypoplasia. Then there can also be persistence in the fetal structures, where the fetal structures since they are fetal structures, meaning they have to stop at the fetal stage, but we can still see them in the neonates. That is an anomaly, and most cases these structures are not functional in the newborns. They will rather bring about problems. So some of these structures can be the foramen ovale, the aortic arch, the atresia, ani. Then we can also have the aberrant structures. Simply, these are structures which will be in the abnormal positions. So, for instance, you can have the heart, instead of being in the thoracic cavity, you might find it in the abdominal cavity. So, such can be observed in malformations. And lastly, these malformations can be duplications of body organs. So you can easily observe that there has been a duplication either of the digits, the limbs, or any other body structure. So the following shall be some of the common developmental anomalies that we can experience in the animals. The first being agenesis. So agenesis is the failure of the organ to develop during the embryonic growth, and this is due to the absence of the primordial tissue. So these primordial tissue, they are like the templates. They are like those tissues from which the organ is in its earliest development. So when the organ is lacking this template tissue, as I can say, it will not have the ability to develop, and therefore we shall miss it in the full development. And this can be observed in limbagenesis, which is also known as amelia. For instance, in this picture, you can see that this calf lacks limbs, the forelimbs. Yes. There can also be agenesis of the corpus callosum, which is that channel that is joining the different hemispheres in the brain, the left and the right. There can also be renal agenesis, where we shall fail to have the kidneys developing. Then we can also have aplasia. Aplasia is the failure of the organ or tissue to develop normally, meaning that the organ will be there, but it has not yet developed completely. And in this first picture, we can see that these dog's forelimbs, they are there, but they are not developed according to the way they should be. And then also in this other picture where we have the pigs, we see that the skin, there's a reddened part. This part has failed to develop. It's part of the skin which had developed earlier, but it failed to completely develop fully. When it comes to atresia, we shall see absence or closure of the natural passage or opening in the body. For instance, there can be biliary atresia, bronchial atresia, vaginal atresia, spina bifida, which is in the spinal cord, then atresia ani, which is that where the anal pore is sealed. We don't have an opening in that side. This is evident in the picture below. When we come to cleft lip and cleft palate, this is where we see openings and splits in the upper lip, roof, or of the mouth, or both, as it is evident for the case of the picture here, where we see that the palate is split. The palate has a split, and then also in the picture of the cat, where we see that its upper lip is having a split as if it was cut. Then we have the prosposus. The prosposus is simply whereby we have a duplicated face. So the parts or the whole of the face can exist duplicated on the same head. Yes, for instance, you can see these curves here. It is the same head, but we can see that it has a different eye, different muzzle. The ears are doubled. Yes, and as well in this cheek we can see the different eyes, the beaks, but they are all housed on the same head, and it is the same case for this cat. Then we have dicephalus. Dicephalus For it, we are having two distinct heads, meaning these are two heads and they are born on the different necks. So this should not confuse us with the prosposus. The prosposus is just the face, meaning that we have a single neck, but we shall see some duplications of the facial parts. Yes, but it is on the same neck. So you can see how different it is in this cheek, in this picture of the cheek having dicephalus, and then also this one having diprosposus. When it comes to syndactyly, we shall see two homodigits in the animal that have been fused together. For instance, in the picture, we see that the calf is standing on one digit, yet it is supposed to be standing on two. With chondrodysplasia, there is failure of the growth plates to develop, meaning the growth plates will not expand according to the way they should, and in most cases, this condition leads to dwarfism. When it comes to cyclopia, which is also known as lobar holoprosencephaly, in this case we shall have two eyes that are fused in one socket. It is well observed in this picture. We have it is a lamp that is having two eyes and they are in one socket. Then we can have brachygnathia, which is also referred to as the parrot mouth. So we shall have the mouth shaped as that of the parrot, the beak, whereby you will see that the lower jaw is shorter and then the upper jaw is longer. Yes, and this is common in the dogs and the horses. And yes, and with the dogs, it will be in those breeds that have short heads. For example, the bulldogs, the pugs, and the boxers. Lastly, we shall have adactylia, where there is absence of the digits, as it is seen in this animal here. We see that one leg is fully having the digits, but then the other leg is lacking the digits, as if they were cut off, but no, this was due to an anomaly where they failed to develop. And then polydactyly, which is also known as hyperdactyly, is whereby the animal has extra digits. So this animal here is meant to possess 5 digits, but we see that it has 1, 2, 3, 4, 5, 6 digits. So this is an anomaly where the animal will possess extra digits. Thank you very much for listening. See you in the next recording.